Where do you want me to sit?
It's recording. Just set it up. Lady was.
Don't look at my mouth bag. You know, I love this bag so much. I bought it there. I bought it for like $50, which is a lot for me. Damage. But this bag is the shit. I'm gonna bring in the car suitcase, man. Because I always bring my suitcases, handles, and fix them. I'm taking back again. But it has 1,500 pockets. If I ever, see me like trying to find my phone right here, insert right here, insert right here. It's by athletic. Tap out left. Hebrew. I love it. Real. It's like a gym bag. If I ever yeah, I'm good.
That's a lot worse because
Some people like my bags. I'm not.
my cover's all over.
But I don't know, just people like they give her things. No, like not like that. I'm not like I like your big cheese shirt. No, I'm jealous I didn't go. I like to go to places where I get.
You got like a shirt or jacket. Oh, that's right there. Okay. Place to work. Hey. Say yes.
Good job.
My mom works there. He's like, I got the whole thing here, and then the jacket. The jacket's like a lumpy. I think it's not fun. My mom works here. I get yeah. No family. Right. Perfect. Get it out. Right.
I was just gonna say, GE's like, he's not like everyone work there. I am. It's like P&G.
Working a big building. And big building. It's a small building. Like every other.
Oh, yeah.
What?
I think citrus is like and it's creepy looking. And Joe's like, I need to
No,
work.
no, no.
He'll always make a joke. Neither day. Or it's like a school for blind or deaf or whatever. And I'm like, you are like great to her.
Her mom is kind of home sometimes.
Yeah, but it just looks like it would be like a school from us here to here. Like that's what it looks like. Yeah, especially like if it's like a storm, it's gonna go down.
And by then she's so tired.
Yeah. I think they probably used to it if they do it more. They don't stay there. They used to be there, right?
Do you used to?
This is in my program, so, yeah.
Me?
Some of the stuff that
Well, I mean, it used to be like institutionalized. Like,
and then I lose track of.
they wanted to simply be like,
Structure.
and then she signed honor and it was all like,
He's always so. Might not know that. I think there's no.
One of the coolest things I've ever seen was like, I was at the park with my sister and my niece. So she was playing in like a dirty stage or whatever. And this
I wish I would
I
have done it this summer.
And then her mom came and like the little boy's mom came right next to us. And she was like, oh, I hope they can play together. My son was like, like after 5 minutes.
It's a lot more used to
Weird language, you know, trying to communicate with each other. And I was like,
I guess I'm worried about people. Is it on a right?
Right, you feel like
when you say like 10 friends, I'm like living in like range.
Is your brother going to study?
I'll study at but I
I was like, okay, that is so sweet.
because you said I make
then I had
Oh, I was 4-year-old. Antoinette just wants to play.
I put something in, I was like, you're not good.
some kids not know how to
Because I remember him in school. I was like, you guys can do the talk about it.
bring it up, bring it up 2 weeks later. Kids love football parties.
We got a present. Swam so much they were going to try fish. But it was still talking about it.
Yeah, like it's so bad.
I was like, you're huge. I was so nervous. Dude, like, you're not going to fish. He's like,
I know.
fish. I'm like, you're not going to fish. Like, I was like, I hate that. I gotta tell her all this stuff. And then I was like, oh, she was terrified. I was like, I'm sorry.
And then I know basic anatomy enough to
What do you say? I'm hungry.
Our bottom.
It's not easy. Yeah, it's the way
There's just so much extra. Joe's sister did it for Jacqueline, brother. And she's almost so just like next to me. She like they'll literally just sit there and pass you on the monitor. She gets up early. She'll sit in the bed. She'll be in there for an hour just by herself. I love it. Playing. She never leaves the room.
Like when it's around 10 years old, I like this is my daily carry-on. The reason I'm wearing it after scarf because I slept in the chair with him.
Good morning, class.
Was actually with the third
Okay, I will give your exams back in lab this week. We'll go through them in lab.
Don't cry.
No, your first exam. And, I didn't think they were bad, but they're pretty good. So move on. Show and tell. Who's got something to share? Ooh, Rosenthal.
Apparently,
Is this going to be good?
it depends on your perspective of good.
Okay.
I've been doing my OB rotation lately, and a lot of pregnant or a lot of women in general have group beta strep.
Mhm.
They test for, and it's really not a problem to the mother, but it can cause issues for the baby when it's born. So during labor,
Right.
we'll give antibiotics
Yeah.
if they're positive.
Yeah, we'll talk in a lot more depth about that. That's Group B, beta hemolytic strep, which is Streptococcus agalactiae, which if you ask that to the head nurse, see if she knows what that means. But the threat is really a meningitis in the neonate if the mother is heavily colonized.
Oh, really?
Yeah.
I was wondering, like, why is it so detrimental to the
Yeah,
fetus and not to the mother?
it's a major problem in terms of bacterial meningitis. And bacterial meningitis can be caused by 15 different bacteria.
Right.
So that's why they will use prophylactic treatment for the mother to reduce the risk of meningitis in the infant.
I was just surprised that they wait until the mother's in labor to give antibiotics.
No, they
Yeah.
give it twice.
They give
Yeah, there are 2 doses. Yeah. One is in the 3rd trimester. And the other one is what, just when she goes into labor? They
Yeah,
changed
right
the
into
protocol
labor.
on that, yeah. Anybody got anything else? That was good. That's the kind of stuff you want to just kind of ask questions about when you see that kind of thing going on, so you have some understanding. We got a notice from the head honchos in the hospital saying if you show any kind of respiratory problems, don't come to work, because we're being pretty much inundated now in terms of people coming into the emergency room, but also staff that are coming into work with respiratory problems because for whatever reason they think they have to work and spreading the wealth. So if you're sick, don't show up, here's what it says. And take care of yourself so that you don't get sick. So that came out. Yeah, that's a good question because I don't know how nursing handles that. And if you've got clinical, they tell you miss clinical, we're going to remove your firstborn.
What they make you think, you're scared.
But on the other hand, if you've got respiratory problems, they don't want you here in the hospital. So I ask them how they work that out. Okay.
a little bubble.
Right. You sit and look.
Give me a full report. There's something that I pulled off my listserv that I was just going to share with you real quickly since we're going to start talking about the viruses today. And that, let me just read some of this to you. About a year and a half ago, there was a series of viruses that were discovered that were huge compared to many of them visible with the light microscope, easily visible, that are now given a couple of names, and there's a big argument now that you can't name things like that, but they call them the Pandora viridae. Pandora viruses because they're so large. And with that, the question is, what's going on with these? So let me just read a couple of paragraphs to you here. A new study by a multinational team of scientists support the idea that the viruses are ancient living organisms and not inanimate molecular remnants run amok, as some scientists have argued. And we'll talk about that today. So the study of giant viruses described in the paper so-and-so and so in the Journal of BNC Evolutionary Biology may reshape the universal family tree, adding a fourth major branch to the 3 domains that Woese came up with. So now they're talking about adding a 4th domain. And Carl won't be around to see that because he died. The researchers used a relatively new method to peer into the distant past rather than comparing genetic sequences, which are unstable and rapidly change over time. They looked for evidence of past events in a 3-dimensional structural domain of the proteins of the viruses. So these structural motifs, called folds, are relatively stable molecular fossils that, like fossils of humans or animal bones, offer clues to ancient evolutionary events. So and so and so and so and so and so. So just like paleontologists, we took a look at the parts of the system and how they changed over time. The parts, what he's talking about, the parts of the virus. Some protein folds appear only in one group or in a subset of organisms. He said, well, others are common to all organisms so far studied. Now they're using the term organism with virus, which makes me a little funny. So he says we make a very basic assumption that structures that appear more often and more commonly in groups are the most ancient of structures. That goes on and on and on. The important thing about this is with this new viruses that have been discovered, and actually in this past year there were 2 more that were discovered. How do we classify these? So it poses a problem. So let's get us into then lecture for today. If you were to go back to your significant events in microbiology, your timeline, and if you don't have it because you threw it away already, think back with me. Let's work together here a little bit. And when you think of the golden age of microbiology, we're starting to equate organism with disease. Yeah, the germ-free disease. Let's find the bug that causes this disease, yak, yak, yak. And so we've got Koch discovering, along with Pasteur, the organism that causes anthrax. Koch discovering the tubercle bacillus that was causing tuberculosis, and so on and so on. And there are just hundreds of organisms being described and then associating the disease caused by that particular organism, if there is indeed a disease. But what was missing about all of those diseases that were looked at? So we find the causative agent for this disease and for that disease and so on and so on. But if you would look at your timeline or use Bowman, that's your text author, and kind of work your way through it, one of the things that you would have noticed so far is that there was no causative agent for mumps, no causative agent found for measles, any kind of measles. There was no causative agent for chickenpox. These are the what? The common...
Viruses.
No, these are the common diseases, childhood diseases, right? So this was a mystery to those people at that particular time. Why can't we find the organism causing these diseases? We can't find them. We take pus smears and so on and so on. We don't see any bacteria, but we know that obviously they're infectious. Whatever is causing the disease, is it just too small that we can't see it under our microscope? So what was the scoop going on here? So there was always the speculation that there's something there, we just can't grow it. We can't grow it on a petri dish with agar in there. It won't grow on that for some reason. So what are these things? And so the term virus, it's a question mark, and there are a bunch of different stories out there. Who first used the term? There are some people say that Pasteur did, who knows. But the term virus starts to be used. And it implies some kind of infectious agent, noxious infectious agent that causes disease. But still, what are these things? How do we grow them? We can't get them in pure culture. We know that we can by taking pus from one individual and moving it to another. We can spread the disease. What's going on? Well, interestingly enough, if you look at your timeline and you look at the end of the 1800s there, And do you see on your timeline a guy by the name of Iwanowski, 1892, yes? 1899, Bajerink. And actually go back to Pasteur, 1885. Pasteur develops a special vaccine for rabies, which, by the way, he didn't do. It was his cohort that really did the work, and he stole that work.
Nice guy.
Yeah, but took the credit for it. So now we're coming up with vaccines for these things. Still, nobody has seen them. We're not growing them in the lab. But Iwanowski and Bajerink are working with what? What is it? What do you see there on the sheet? They're working with tobacco. And they're trying to study the disease that is destroying large numbers of tobacco crops, which they refer to as tobacco mosaic disease. Mosaic is what? Yeah, and they're put together in little pieces to make a picture, and so you get this blotchy kind of a thing until you finally make some sense out of it. So the question is, well, why tobacco? Why the big interest in tobacco? Well, tobacco is a huge money crop, and it still is a huge money crop. So if, whether in the United States or whether you're in Northern Africa or whether you're in certain regions of Europe, if you had tobacco, you could make big bucks. But if the leaves on your tobacco, you couldn't sell at market because they had this mottled look to them, this mosaic kind of a look to them. You couldn't sell your product, and your family went without. So they kept saying to the microbiologists at the time, come up with an answer, find the bacterium that's causing this disease, because it's got to be a bacterium, and let's make some sense out of this. Well, what those two individuals did, basically, if you look what it says about Iwanowski, first to isolate a virus and show that it can be transmitted in a cell-free. What he did, he still didn't see anything. All he did was extract plant juices. So where it says that he purified the virus, all he did was simply concentrate these things that he couldn't see, but he knew were there, in this fluid, plant extract fluid. That was all he did. If you look at Bajerink, what did he do? He further elucidates, great word. He stretches this out to say that there's a viral agent of tobacco mosaic disease, and postulated that the virus has many of the properties of living cells, and that they can reproduce within cells. God bless you. So now we're starting to describe something no one has seen. And it makes it kind of interesting out of all of this. And then if you jump, let's just do a big jump here, into the 1900s, and let me just see if Stanley is on here. Anybody see Stanley? You may not be on.
1935.
1935, there it is. So what did Stanley do? What does it say?
Using tobacco mosaic virus to form crystals.
Okay, so people are still studying this virus, whatever it is, and they're just using that term, tobacco mosaic virus, because when they try to take the filtrate and put it on other plants, it doesn't cause a problem with those plants. It seems to only infect tobacco. But Stanley then, working with it, discovers that he can crystallize it. So then the question is, wait a minute, you're crystallizing, the living cells don't crystallize. And you know, salt's a crystal, right? You know that from chemistry. You put two things together and you end up with this crystal. Well, living cells don't crystallize. So the question is, what's going on here? So let's begin to put some pieces together. We know we've got something that causes disease. We know that it's infectious because you can take an extract from this plant that's diseased, add it to another plant, and it gets the disease. We know that humans can get diseases that we can't see, so we got something there. And by the 1930s, then something else occurs. We start to chemically analyze the viruses, chemically analyze them, and what we see is that they're made up of protein and some kind of nucleic acid. Now that nucleic acid can either be DNA at that point, or RNA. And at that point, they said it had to be one or the other, but not both. But now we realize that there are exceptions, as there always are in biology and in the sciences. There are always exceptions, that there are some viruses which do have both. And those were discovered actually rather recently as well. But for our sake here, let's just kind of tunnel vision ourselves and say that the nucleic acids are DNA, typically, or RNA, but not both. And there's protein there. So now we're talking about organic compounds, but yet we can crystallize them, and yet they seem to replicate. So let's put the pieces together. Let's try to do that. Well, as we move through the 1930s, and Stanley's doing this, and people are still scratching their heads in terms of what we've got here, nobody has seen them yet, we finally have the development of the electron microscope. And with the electron microscope, for the first time in the 1940s, we get clear pictures of these viruses. And what we see with these is that they take on a variety of shapes or forms. Some of them can be round-like, some of them can be cylinder-like, and not to confuse that with a bacterial bacillus. Some of them are just kind of irregular and filament-like. Others take on very distinctive, almost gem-like forms that we commonly refer to as the icosahedrals out of all of this. So we now realize that we do have something there. We've got a particle of something there. We know that it's made up of organic molecules. But the question still is then, well, what do they do? And how do they work? We know that they're associated with disease, but how do they do that? And do we have specific viruses, or is it just one kind of virus out there? So all these questions had to be asked. And we don't start to really answer those questions until the 1950s. My point of all of this battling on here is that this is not long ago. Everybody with me? 1950s is not a long period of time. So one of the things I've tried to stress to you in this course is that microbiology, compared to the other sciences, is a very young science. And it's still kind of, we're knowing stuff all the time, finding new stuff out of this. So now we're going back to, 60 some years ago, and we're finally beginning to put the pieces together. So what do we have here? Well, in terms of the protein, what was seen was that the protein now makes up this kind of outer wall, whatever that happens to be, which we're going to refer to, and it's not a cell wall, it's referred to as a capsid. And it's made up of proteins. And especially with the electron microscope, we were capable of looking even more closely at that and seeing that this capsid is made up of smaller, and it's a protein, made up of smaller proteinaceous units that were called capsomeres. It's all in your textbook. Capsomeres. So the analogy, if we had drawn an analogy, looking at this room, and this room is one large virus. And inside, we're in here, and we are either RNA or DNA. And the walls, the floor, the ceiling, all of this is made up of this structured protein that we call capsid. But if you could envision, how can we do this? That the entire room, all the walls, ceilings, and floor were made up of the same cinder blocks, cement blocks. They have a distinctive shape. They're all identical to one another. We simply put them all together. This represents the capsomere, this smaller protein unit, and you've got identical capsomeres then that go together to produce this capsid. Yes?
Okay, so Stanley was the one to discover all?
No, Stanley discovered that he could crystallize. And the question was then, wait a minute, living cells don't crystallize. But he was capable of crystallizing this by actually allowing proteins to link together. So you beat me a little bit to the punch here. The next question would be then, what Stanley's work, how does you make sense out of that compared to what we now know? And the thing is that these unique capsids, these protein covers on these viruses, fit together like pieces of a puzzle. And you could crystallize these based on the structure of the protein involved. And again, we don't think of that usually in terms of biological cells. They don't crystallize, but these did because of the unique nature of this outer protein coat. So inside this capsid, whatever it happened to be, whatever it looked like, inside this then was going to be this nucleic acid, which could take on then a variety of forms. Now, 1950s again, what do we know about DNA? What's happened? We know it's there.
Well, they know like the structure, like double helix structure.
Okay, when do we really start to see that? Watson and Crick did their work in the late 40s and they published in the 50s. So now for the first time we get an understanding of that. And so we see a molecule then of DNA, which DNA is a double helix. It's got two sides to it. And then what comes next is that RNA, we start to look at RNA and we look at the structure of RNA at that point. But what they find with the viruses is that we can have all kinds of scenarios involved then with these nucleic acids. So we can have the typical double-stranded DNA, which we're used to, that double helix. We can have single-stranded DNA. We can have triple-stranded DNA. We can have RNA, which is single-stranded or double-stranded, positive or negative. The point I'm trying to make is that the DNA or RNA, which is housed inside the capsid, protected by the capsid, depending upon the kind of virus, can have a lot of variety. I shouldn't say it, that implies that can change. A distinct virus always has a given kind of RNA or DNA, but it can be of a whole variety of different forms. So this complicates the matter again. So if you've got something which has got RNA only in it, how do you explain how this can replicate and can metabolize, or does it? So the questions keep piling up through the 1950s. We're trying to put the pieces together, and it's not making a whole lot of sense at that point. A couple of other things from a anatomical point of view is that this nucleocapsid, as it's sometimes referred to now, nucleocapsid simply means that we've got this protein coat with some kind of nucleic acid on the inside, is that sometimes that nucleocapsid, if this is a virus up here, has an envelope around the outside. An envelope. And some viruses don't have an envelope. And then the question is, well, what is this envelope constructed of? Chemically, what is it? So we analyze the envelope and we realize it's a biphospholipid membrane. Okay, let's just put that under our belt. So those viruses that do have the envelope, we simply refer to as enveloped viruses. Those that don't have an envelope, we simply refer to as being naked. So think of this for just a second here. We're starting to put pieces together. And we're looking at possible variations based on the structures that you see. God bless you. Again. So now, when you start to see these variations, we can begin to come up with a means of classification, can we not? So we can say, okay, what is the shape? And we'll use that. Does it have RNA or DNA? What kind of RNA or DNA does it have? Does it have an envelope? Does it not have an envelope? And we start to put viruses in groups based on similar characteristics. But what's not going on here is an analysis of the genetics of the organism to say, are these related or not? Plus the fact that we've got a whole other group of budding virologists who are categorizing these viruses not by so much by their physical characteristics, but by the diseases that they cause. And I'll show you where there's a problem with that. So hepatitis viruses, those viruses that cause hepatitis, liver problems. Initially, they took all of the viruses that cause hepatitis and they put them into one bag. With further analysis, what happened was that we realized that they're very different. They just all seem to infect the liver, but they have no of these other characteristics common to them. So that didn't work, simply using the disease as a method for classifying these. We realized that hepatitis A, B, C, D, they're all over the place in terms of what they're made up of. So obviously they're not genetically linked to one another. The next question that was asked then is, what do they do? So we know that they invade cells, specific cells, and what do they do when they get in there? Well, they see a variety of things happening. In one case with the tobacco, what happened? We simply took the leaf and somehow the cells in the leaf started to reproduce abnormally, and we end up with this abnormal tissue, which was called mosaic. We know on the other hand that if you get a chickenpox virus, what does it do? It spikes you a fever. It has these pustules that break out all over your skin, and so on and so on. So what are they doing? And obviously they've got to reproduce. How do they do that? So one of the terms that starts to be used during the 1940s and 50s was referring to viruses as obligate intracellular parasites.
So when, sorry,
Go ahead.
so this was in like back, in the 1800s that they did they discovered that it had protein and RNA or DNA?
Actually, most of this was done in the 1900s, in the 20th century.
Okay.
So again, to try and, if I'm getting you confused, let's just look at the timeline again. 1800s, golden age of microbiology. And one of the exciting parts of that time was to associate an organism with a disease. This bacterium causes this fungi causes this. But they also had a slew of diseases for which they could not assign an organism. And at the same time, so while they're trying to say we just got to find some way of finding what the causative agent is for all this, they're also looking at that same time period, some individuals looking at tobacco because of the economics of the crop. And so we got kind of two things going on at the same time. They thought that they either had a fungus or they had some type of a bacteria that they just couldn't grow, and it was causing this particular disease. The two pieces were not going together at that time. So we got plant pathologists doing their thing in the 1800s. We've got the bacteriologists doing their thing over here, not knowing that they were going to link together. So we move our way finally into the 20th century, into the 1900s. Still haven't seen a virus yet, but we know that something is there. And then when Stanley figures out that he can crystallize this, and now we know what the chemical makeup is, none of this makes sense. We finally move into the 1940s and we can see them finally. So we know that something is there. And then we're starting to put the pieces together. Okay, what do we have? We've got some type of a protein coat around the outside. We're going to call it a capsid. It's made up of identical repeating units that are glued together. We're going to call those capsomeres. In the middle of that, no cytoplasm, no nucleus, no organelles. We're not finding any of that crap. All we're finding is DNA or RNA and some enzymes typically. But nothing else there, no ribosomes. So you got protein, but then your question comes, the only way you're gonna get protein is if you send a message to the ribosome, messenger RNA, all of that crap hole, and you pull on amino acids and we make them. None of that stuff is there to allow that to happen. So during the 1950s is when we just start to put the pieces together. Is it helpful?
Yeah.
Okay. So now we're referring to these as obligate intracellular parasites. Obligate means what? You're obligated to someone. You owe them something, right? You got to be there. And in this particular case, these parasites have got to find a host, and they've got to enter that host, a very specific host, to cause whatever disease they cause. They're obligate intracellular parasites. So what do we have then? Let's take a look. Sure, let's just use this. No yawning. So what you see here what we refer to then as a naked virus with its nucleocapsid. Got a nucleic acid of some kind on the inside, got the capsid on the outside. And you take a look at the virus on the right side, and it's an enveloped virus. And if you look at it, it looks like this same virus in here, but with this structure around the outside, referred to as an envelope, which looks like a biphospholipid membrane. What you notice as well, if you compare the two, and again, we're starting here in a very simple kind of description of all of this, is that if you have an enveloped virus, it also has proteins which stick out through this envelope. They go by a couple of names. The old and the original name was a spike, and we'll simply refer to them at that point. So if we're taking a look at the anatomy and physiology of these particular organisms, the question then is, okay, I see an anatomical structure, there's got to be a reason for it being there, and you'll see in just a couple of minutes what that is.
The spikes, are those secured to the capsid?
They actually come from the capsid, they come all the way through, and they're an extension, a proteinaceous extension of the capsid, and they stick out through the envelope. During the 1940s and 50s again, there was another type of research that was going on that dealt with infectious agents of bacteria. And this was kind of an eye-opener for the bacteriologists because they realized that these viruses didn't just attack plants and animals and humans and things like that, but they also could attack bacterial cells. So nothing was free of a possible attack by these organisms. And what they found as well was that the typical virus which attacks a bacterium has this unique shape. So if you look up at the top, it looks like our sample that we just showed you over here. And that icosahedral type of a formation with a nucleic acid on the inside. But then they saw that there was a whole lot more structure to this. And it looked a little bit like a lunar landing module. We were on the moon once. A couple of times. A bunch of times. Everybody knows that, right? No.
NASA is trying to get commercial companies to sponsor a trip back to the moon.
Yeah, because we don't support NASA anymore, so they've got to go to outside support. So the question then was, okay, we got this goofy looking thing, and here's the electron microscope photograph of that, and it looks pretty much just like that. So now we're actually being able to see these by enlarging these about 1 million times out of this whole thing. So it made sense then, in terms of trying to understand how viruses work, as opposed to using human tissue or animal tissue or plant tissue, let's work with bacteria. They're easy to grow in the laboratory. and we don't care if they die. And we can, because we have, billions upon billions of them in a test tube, let's see if we can make some sense in terms of what the life cycle is of a virus. So with that, let's just do this on the board.
At least people not know about getting infected because viruses are so
A lot of them did. And you'll see in the video I'll show you in the second part of today how we've come a long way in terms of being careful when we deal with the viruses. So what they found with these viruses that attack bacteria, and they gave them a unique name, which is what?
Bacteriophage.
Yes. The bacteriophages. Bacteria, that makes sense as a prefix because this is the host. Phage means to phagocyte, wandering macrophages, to eat. So we've got, and they're not eating, but that's the term that was used. So we've got unique viruses which can, as it were, eat bacteria. And the cycle that was set up after looking at this time after time and being able to follow these along with the electron microscope, and at the same time we were also introducing some radioactive compounds into the protein so we could track them along, is that here was a bacterial cell, and it was specific. So we got a specific genus and species of bacteria. The phage, which is floating around all over the place, happens by chance to find a host cell that it can attach to. Okay, so what's going to allow for the attachment to occur?
The tail fiber.
Okay, so we got these little tail things coming out, but what would you suspect is going to allow for the joining of this bacteriophage with this host cell?
Outside?
Somebody said, yeah, and receptors. So there are specific receptors that are found unique to this species of bacterium, that it's not out there flagging down bacteriophages going, hello sailor, it's not that kind of thing. What we've got is they're using them for something entirely different, but this bacteriophage is capable of attaching itself to those proteins. It's lock and key kind of thing. So now we have the virus which is attached to the host cell. And that goes by a variety of names. Sometimes that term is called adsorption. Sometimes it's called simply attachment. But we have virus linked to host cell. The next thing that they noticed happened when it was unique to bacteriophages is that when they looked next in the sequence, the bacteriophage looked like it had just squatted. And what they noticed was that it had punctured the cell wall and the membrane and had introduced its own RNA or DNA, whatever kind of virus it was, into the host cell. That's called penetration. So our step one was simply to attach. Step two, in this particular case, was penetration of the nucleic acid. That's the only thing that went in. And perhaps some enzymes. Now the question, well, where did those enzymes come from? And hold on to that, and we'll tell you in just a second here. So penetration of the nucleic acid plus any enzymes that this particular phage was carrying with it. Yes?
Okay, so the bacteriophage thing,
Mhm.
that's what it's called, and what is it attaching to? I want to get it
Okay,
right.
this is the host cell. This is a host bacteria. Here is the virus, which was free until it found attachment to the host bacterial cell. It attached or adsorbed onto the surface. It then introduced its RNA or DNA, whatever it happened to have. That's called penetration. So now we've got viral RNA or DNA inside the host cell.
What are the things it attaches to called?
Receptor.
Yeah, there's got to be some kind of a protein receptor found on the cell wall that's going to allow, or the cell membrane that's going to allow for a specific virus to attach to a specific host. Yeah.
And what's the second word under resorption?
This is attachment.
Oh, okay.
I'll take my time and print more clearly. Yep.
It's on page 383. This one.
Yeah, the whole thing is outlined in your text. So now we've got this nucleic acid, viral nucleic acid on the inside. Let's assume in this particular case that it's DNA. Well, if it's DNA, that means it's also going to have a set of genes along it. And the genes that are found there, in turn, are genes which are going to code for new capsomeres. Makes sense? And for enzymes that allow for the capsomeres to be put together, and for enzymes that allow for us to complete the whole project of the whole thing. So what's going to happen then is that we're going to start to synthesize viral parts. So we're starting to build the pieces of a new virus. And not just one, but a boatload. If the virus was an RNA virus, to keep the story simple, instead of having to go DNA, copy with messenger RNA, go to a ribosome, and all of that, the RNA functions typically as messenger RNA, meaning that we don't need the DNA. It's as if it had already been copied from the DNA. The end result, though, is that we're starting to synthesize viral parts. We're starting to make the capsids. We, in turn, are starting to copy the RNA or the DNA, whatever it happened to be. And we're also producing viral enzymes that allow us to put all this together. After we have gone through the synthesis of the parts, step 4 would be assembly. Let's put the pieces together. So now we're assembling all of those pieces until, what do we have inside the host bacterial cell? A whole bunch of these new bacteriophages. And last of all, what is this virus going to do? Produce some kind of an enzyme which kills the host cell, and all of these escape. So what would you call step 5?
Release.
We'd call it release for. So for the first time, we had some kind of an idea in terms of how viruses do their thing. Now, what you will notice out of this is that the virus came in with next to nothing. Basically, its own nucleic acid, RNA or DNA, and maybe some enzymes to get the whole thing started. But it depended totally on the host cell to do what? Supply it with the amino acids to make the capsomeres, with any ATP that had to be there. The host cell had to supply the ATP to get it all done. If we're going to replicate any of the RNA or DNA, the host cell had to take care of doing all that. So what's going on here? The host cell then becomes what? The servant of the virus. And what does the virus do after the whole thing is over? It kills the host. And we start to use some other terms at this point. One of which is, how long did it take for this to occur? From the time of attachment or adsorption until the time of release, what they saw was with specific viruses that there was a specific time involved. And they referred to that then as burst time. How long did it take for this cycle to occur? And what they also noticed was that typically the same number of viruses was produced, mature viruses that got released at the end. Depending upon the kind of virus, the species of virus, it attacked this host cell, and the host cell produced a given number of new bacteriophages. That was referred to as then the burst number.
What was the burst number?
The burst number tells us how many viruses, new viruses, are going to be constructed by this host cell infected with this very specific virus. So these become species-specific characteristics. Now we're using the term species, though, with something that's not living. Because is a virus living then? You know, in your text on page 397, it's got this comparison of viruses and viroids and prions and real-life cells. And so sometimes take a look at that. So what are we missing here that would be typical of a real cell? We don't have cytoplasm, right? We don't have a cell membrane. But then you go, wait a minute, there was this outer envelope, but it's outside the capsid. Doesn't seem to have the same function that a normal cell membrane would have. It doesn't do anything, does it? It doesn't metabolize.
So does it kind of act like a spore, just waiting for the right thing to?
Do not use that term, ever again.
I was thinking the same thing.
Oh, geez, you're
Like,
messing.
yeah, it sounds like,
Yeah.
it's got the
Don't
same.
go, don't even wipe that word spore out of your memory bank. What we have here is the unique parasite, right? Because it depends upon the host cell to do almost everything for it. What?
The virus just comes in and uses up all the host cell stuff, and then the host cell dies, and then the virus is replicated.
Exactly, and so now we start with one coming in. We may produce 15 new phages. They're going out and attacking, you know, another, within a 30-minute period of time, another 15 bacterial cells. And look at all of a sudden, and then you go, wait a minute, if this were the case and every bacterium has a phage which is specific to it, there wouldn't be any bacterium left. Right?
How does that work? You can't, cure the virus with medicine, so.
So we've got a question to answer, don't we? We do. Yeah.
So is this the same as the disease process where you start off with a cell that, I mean, the way I'm thinking about it is like cancer or anything else, that it kind of does that same kind of replication?
We can draw some analogies from that, but you brought up the word cancer, and we're going to hold on to that one too, because we're going to see now when we start to take a look at animal cells, how did we get cancer then? How does that occur when we're dealing with these?
But what about AIDS? Or, I mean, is it the same thing, the same process?
Yeah, because that's a virus too. So now let's take a look at this. Before we bring down. It's a little small, but we'll walk through it together. Okay, I'll give you a copy of this. There's something similar in your text, but I like this one. So we have two viruses here, virus A and virus B. And what you can't see up here is that virus A is an enveloped virus with its spikes sticking out. Virus B is a naked virus. So what happens in both cases? They're attacking not the same cell necessarily, but they're attacking a host cell. And we have the first step there, adsorption or attachment. And what's happening on this side is that, now they use the term engulfment. What did we use for that? Penetration. So you see different terms used around here. But what's happened is, what has the host cell done? It's been irritated by this, and we're going to see loads of variations on the theme. Actually, what could have happened is that out here, this viral capsid could have stayed on the outside, and it simply dumped its RNA or DNA inside. Same thing over here could have happened either way. In this particular case, the host cell has endocytosed. And, big mistake on the host cell's part. Huge mistake, but it doesn't know any better. It's like having body lice. How many of you have had body lice?
Oh, body lice.
Body lice.
Ew.
Okay. Some of you have them right now? How's your ear doing, by the way? Does it look good? So now, we've got this irritant on us, and they take it in thinking they're going to kill the thing. But what does it do when it gets inside? It uncoats, releases its RNA or DNA, and with that, get over here. Now we've got, and in this particular case happens to be an RNA virus, now we've got the viral RNA inside the host cell. Well, there's no turning back at this point. What is it immediately going to have the host cell do? It's going to function in this particular case like messenger RNA, and the host cell is going to start to read the genes off of that, the gene units off of that messenger RNA. And what does the host cell start to do? Produce proteins. What are those proteins? Some are enzymes, some of those are capsomeres. So we keep going. The host cell is now producing more and more and more and more of these things. And now so what we're doing is synthesizing. And in this particular case, let's follow two scenarios up. If it's an enveloped virus, what happens is we're starting to now produce these spikes. And look where the spikes begin to show up. On the host cell's membrane. So under the microscope, we can look and go, we've got a cell that's abnormal. You can see the cell. We know that the viruses are inside. So it's displaying certain kinds of characteristics that indicate that this cell doesn't have long for the world. So following this up now, we go through assembly. There are all the nice little viruses, except that we want this to be what kind of virus? An enveloped virus. So what's going to happen next is the host cell, in turn, as you can see right there, starts to take these viruses and pinches them off. And it's called budding in this particular case. And with every intact, complete virus that it pinches off, what does it take with it? The host cell's membrane with the spikes in it.
So what does the host cell do after those things are gone?
We got some options, and one of them would be that in some cases we'll find what we call latency that occurs, is that this cell may not die right now, this host cell. It may stay alive for years and years and years, but every once in a while what does it do is it releases some more viruses. Chicken pox.
Shingles, plague or not.
So what you've got is you always have the virus once you've got the chickenpox virus.
Which is good, because it protects you, right?
From what?
Getting reinfected.
Getting it again.
Yeah, but when you get older, you get shingles.
Yeah, well, I mean, I thought that was like, because you got penetrated.
No, it's the same virus.
Virus later.
Same virus you picked up, and it goes into latency.
Okay.
So now we've got this host cell, which may live for a long period of time. So does it have a burst number or burst time? No. But what happened on this side? We did the assembly. There are no spikes in the membrane because it was not a enveloped virus to begin with and doesn't have the genes to call for the production of those proteins called spikes. So what happened in this case is we simply produce our given number, our burst number. What does it do? Kills the host cell, lyses the host cell in a given period of time, hence the term burst time. So a difference then between enveloped viruses and naked viruses in terms of how they do their damage. The thing to keep in mind here is that who's doing all the work here? The host cell.
So is one worse than the other one to have?
No, it just depends upon the particular virus that you've got. And some of them both could be lethal. They could kill you.
But do enveloped viruses still have a burst number?
Enveloped viruses do not.
They don't.
They have no burst number and no burst time. But the virus goes into something referred to as latency. And latency implies what? If you're latent?
No, it just means that it just hangs on.
Yeah, you're waiting. So they can pinch these off. And you know, what might this cell do? It might be stimulated by stress, all kinds of other factors, and dump out a whole bunch of these. It may just lie latent for 80 years, and all of a sudden decides it's going to start. It's been reproducing along. Every time it reproduces, it does what? Reproduces the virus along with it.
Is that herpes?
Yeah, exactly. Herpes is another great example. The gift that keeps on giving.
So I have a question. Because one's naked and one's not, and like the one that's not naked has a phospholipid bilayer, right? Is that what we're talking about? Has like you know what I mean? Has the little outer shell, has that protection. So because one has protection and the one doesn't give that protection to the host cell. That's why sometimes it doesn't die. Like,
Yeah.
what makes one different?
It just has to do with its own genetics. What genes does it have on its viral RNA or its DNA which allow it to function in this particular way?
So there's no reason that it has extra protection. Does it like make it harder to die, or it just no?
No.
It just has it.
One of the problems, though, is that, for instance, with HIV, and why do you not say the HIV virus?
Because we're so sensitive.
Okay, because you're stuttering then, right? The virus. Is that it's an enveloped virus. And one of the things that it does when it destroys tissue in the human body and proceeds to move on is that it carries with it a lot of markers off of that original cell, so that sometimes our immune response to that, whether it be B cell or T cell response, is that it feels around and goes, these are spikes here, which I don't think are my proteins, but it's got all these other proteins which are mine. And so then your immune system goes, I'll just take a step back and wait to see what happens here. And of course what happens is the virus spreads like wildfire in certain tissues, and all of a sudden the immune system goes, I think I better deal with it now, and it's too late. So it fools, it's a trickery. But it's not necessarily, having an envelope does not necessarily mean it's going to have this huge protection. It's simply a mechanism that it uses. Yeah.
Doesn't HIV specifically attack the immune system?
No.
No?
No, it'll attack mucosal cells, it'll attack neurological tissue. Yes?
I mean, that sounds like a lot like when we go back to my son's cancer, because he had it for 5 years before it even showed any signs. And then we took all this immunotherapy just to hold it back because we couldn't kill it. And then 18 years later, that's when it showed back up in full force. So it sounds like it was it was doing this. It was just.
There are a lot of oncogenetic viruses. And we'll talk about those actually to kind of show you how that system works. But you can easily go into a latency period, easily, with that. It's not uncommon at all. Yeah.
Is this the same process for like Bell's palsy?
Bell's palsy is a viral disease of the trigeminal? Yeah, and it's the same kind of scenario where it stays in latency for a period of time. And individuals will have flare-ups and then it goes away. And then flare-ups and then it goes away. I'm trying to remember which of the trigeminal which of those is most severely infected. Does anybody know? Do you know? Yeah. Find that, because offhand I just don't remember. Okay, let's take a break here. We're around 15, so get out of here. Start. it the facial?
I thought so bad.
Yeah, that would make sense.
Yeah,
Yeah.
the facial.
Yeah.
Public in America.
Hi there.
List the virus. Sure.
Specific, so
Very specific.
would it be possible to like engineer viruses to attack users?
Exactly, and we'll talk a little bit about the whole idea of gene therapy, viruses. We can manipulate them so that they don't destroy necessarily the host cell, or can destroy a host cell, and use them as a mechanism for, in one form or another. We'll talk about that a lot. But